नमस्कार, सुस्वागतम, केम छो, आदाबार्स, गुड आफ्टरनून। मैं आप सभी का पीआरएल के अमृत व्याख्यान में स्वागत करता हूं, अभिनंदन करता हूं। ए वेरी वार्म वेलकम फ्रॉम मी अनिल भारद्वाज फॉर पीआरएल का अमृत व्याख्यान। टुडे is the 45th Vyakhyaan of the series of Vyakhyans being organized by PRL as a part of PRL's 75 years of legacy and history in fundamental physics and space sciences. Established in the year 1947 by the father of Indian space program, Dr. Vikram Sarabhai, the PRL's Platinum Jubilee coincides with India's 75 years of history of independence. Hence, it's a joint celebration of the development of science and technology in India by PRL under the banner of PRL का अमृत व्याख्यान. Today, we have yet another very distinguished Vyakhyaan or the speaker today, Dr. Vandana Prasad, who is the director of Birbal Sahni Institute of Paleosciences in Lucknow. And she is going to talk on role of planet tectonics and climate in the evolution of tropical angiosperms, evidences from Indian fossil records. We thank Dr. Vandana Prasad for agreeing to our invitation and joining us on Platinum Jubilee celebration of PRL. I would now request my colleague, Professor Deshpande, to kindly introduce our today's Vyakhyaan Karta to our audience on WebEx, as well as those who have joined us live on the YouTube. Over to you, Dr. Deshpande.
Thank you very much, Professor Bhardwaj, and good afternoon, everyone. It's indeed an honor for me to introduce today's speaker, Dr. Vandana Prasad. Dr. Vandana Prasad is currently the director of Birbal Sahni Institute of Paleosciences, BSIP, Lucknow. She is a highly acclaimed paleobotanist from India and well known worldwide in professional circle for her landmark contributions in paleosciences, which has immensely advanced the understanding in botanical aspects of earth sciences in India. Her landmark research includes discovery of earliest grasses some 65 million years ago from India, which was published in Science, and the origin of 65 million year old wild rice from India, which was published in Nature Communication, and evolutionary history of sal forest of Southeast Asia, which was again published in Science. Her other well-cited publications deal with paleosciences, including the 55 million year old Gondwana relic rainforest in southern Western Ghats, early Paleogene extreme global warm events from Indian sedimentaries, effect of Deccan volcanism at Keti in Meghalaya, sequence stratigraphy of Vastan lignite, and role of mid-Holocene climate on the Harappan civilization, so on and so forth. She has several such important research contributions to her credit, numbering in excess of 50. She has received several awards and honors at national and international level. Once again, to name a few, she was the recipient of a fellowship under INSA Exchange of Scientists program to work at Bonn University in Germany. She is the recipient of Smithsonian Fellowship at Smithsonian Tropical Research Institute, Panama. She was invited by the Indo-US Science and Technology Forum to participate in the Indo-American Frontiers of Science Conference, as well as Indo-German science collaboration to contribute in the environmental challenges in Asia. She was also invited by the Institute of Geology University Louis Pasteur in France to carry out palynofacies analysis with a sequence stratigraphic framework. So, you can see that she is very widely acclaimed in her field. She is a fellow of the Indian Academy of Sciences, and at national level, she is associated with various review committees of different government departments. We are indeed very happy that today we have with us such an illustrious speaker. So with this brief introduction, on behalf of all of us here, I warmly invite Dr. Vandana Prasad to please deliver her Vyakhyaan. Over to you, Dr. Prasad.
Thank you, Dr. Deshpande, and good afternoon, everybody. I would like to thank Dr. Bharadwaj and for inviting me to this function. And it's a pleasure to be part of Platinum Jubilee celebration of PRL. So I am very thankful to Dr. Bharadwaj, and I thank Dr. Deshpande for inviting me for this to deliver the lecture in this function, and all the organizing committee members who are supporting this this function. Thank you so much. So let me share my slides with you. So before I start talking about my work, I would like to talk about the institute. This institute was established in 1946.
Vandana, this slide
Professor.
is, excuse me, this slide is not visible actually. Your slide is not visible. I think some another screen is visible. Maybe you need to click the window in which the PowerPoint was there. We are seeing your screen now.
I think there is some issues.
Bushit bhai, can you suggest something?
PowerPoint slide is closed, I think.
I suggest that please unshare and start sharing again.
Yeah.
Yeah.
In fact, that file is not open.
May I again join? I don't know what is happening.
No, actually, Bushit bhai, can you unshare please from your end?
I can.
So that she will share once again. Yeah. Now you may please open this your PPT first, and then maybe it will work nicely.
It says you can't share content unless the host, a co-host, or presenter makes you the presenter.
You are the presenter now. You can try again.
Is it okay?
Yeah, it's okay. Now you can make full screen and close this. Yeah.
Perfect.
Yeah, it is fine.
Yeah.
It is fine.
Yeah,
Absolutely
it's okay?
fine.
Yeah,
Yeah,
okay.
please.
Okay, so just before I start my talk, I would like to talk about the Birbal Sahni Institute. For those who are not familiar with the institute, the institute is an autonomous institution under the Department of Science and Technology, and it was established in 1946 by Professor Birbal Sahni to carry out paleobotanical studies. Over the years, it was felt that fossil studies needed a push with emphasis on modern thoughts and technology, and this idea of expansion led to the renaming of the institute from paleobotany to paleosciences. And with that, the mission, our mandate of the institute has also been changed. The mission is to study the various aspects of paleosciences, the drivers, impacts, and processes to provide models that are different in today's world to understand past climate, paleobiodiversity, paleoenvironment, past civilization in order to increase the credibility of future climate change projections. So now coming to my topic, the role of paleotectonics and plate tectonics and climate change in tracing the evolutionary history of angiosperms, evidence from Indian fossil records. So when we talk about the angiosperms, that we can see that the angiosperms, they evolved during Triassic time period, late Triassic, around 230 million years ago, and it was not in the Gondwana continent; it was in Laurasia. So these continents, they were in the tropical region, and there are some records of angiosperms that tells us that angiosperms started evolving during the Triassic time period. However, many scientists, they found that there is ambiguity in these fossil records, and they are not true angiosperms, so they are known as early Triassic stem angiophytes. After the Triassic, then there is a gap in the evolution of angiosperms. There are very less records during Jurassic time period, and probably because during Jurassic time period it was mostly the global climate was arid. So we all know that angiosperms, they are moist-loving plants, and so their fossil records are very scanty during Jurassic time period. Then, as during the early Cretaceous time period, we see a sudden radiation of angiosperm records. And angiosperm records started appearing in early Cretaceous time period, and slowly by end Cretaceous, there is a full expansion of angiosperm fossil we can see across the globe. So we can say that angiosperms, they came very late as compared to gymnosperm ferns, cycads in the geological time span. Once they arrived, they completely cover the globe, and they could compete with other plant groups. So what are the features that make them very successful in the evolutionary aspects as compared to other plant groups? So before that, let us talk about what are the early land plants. So we know about the Glossopteris flora. Most of you people must be knowing about that Glossopteris because they are all the Permian coal that is found in Central India and eastern part of India. They belong to Permian coal, and this coal is derived from the Glossopteris vegetation. So there are some features which are the scientists, they say that there are some features which are they can be precursors for angiosperms. Apart from that, then William Soniela, that is also, and the species now are extinct, but they have some features which are also found in. So it is considered that they could be the precursors. Let us talk about the biological traits which are peculiar, and because these just they can be ecological, they are ecologically, and they seed in the evolutionary history. Morphs are the presence of carbon is a protective structure that covers the female gametophyte, and then the flower structure. We know that angiosperms, they are also known as flowering plants, and the flower structure which is typical in angiosperms, bisexual characters, that is both male and female in the flower, that makes that because of the fertilization can be possible. Double fertilization system, which is peculiar in angiosperm, and that also helps in the growing angiosperms, growing embryo. Then cross-pollination mechanism, which is also found in angiosperm, which is also a very, very advanced feature, and that is also limits the inbreeding and maintains the genetic vigor. Nectar production and pollination through different agents, which is also is found in angiosperm, and that makes them some advanced characters, and they make them succeed over other plant groups. Fruit and seed disperser, we all know that fruit and seeds, they are mostly what we eat. Edible fruit and seeds, they are produced from angiosperms, and because the animals or insects, they eat them, they help in the dispersal of angiosperm to a very far away places. Some ecophysiological traits are xylem vessels, which are present in angiosperms. They do the effective water transport to a the plants which are very high in length. And the two characters, leaf venation and stomata, which are very, I'll talk about in detail about these two characters. The leaf venation, which is, if you compare the leaf of angiosperm and gymnosperm, they are very, very different. In angiosperm, the venation system is very complex. It is very reticulate, and we can see that it's a very intricate. And so leaf, we all know that the plant do transpiration. The plant also provide water vapor in the form of when they transpire. So the leaf venation, they help. The more the leaf, having a leaf venation system, it will help in the more water transport. So there are some a group of scientists. According to them, from the start of early Cretaceous, the climate started becoming very humid. And they say that angiosperm, because they evolved that time, and because of the main function of transpiration in angiosperm, and they transpire, and they put water vapor in the atmosphere, the climate become very humid. So this work was done by a group of scientists that angiosperm helped put the rain in the rainforest. Without the angiosperms, the climate would have been very hot and arid according to them. Similarly, the stomata, we all know that the stomata, they help in the gaseous exchange. They help in the photosynthesis. They also do the transpiration. They also do the respiration also. So stomata, if has been found that the carbon dioxide, if it is very high in the atmosphere, the plant has the capacity, angiosperm plants, they have the capacity, they have the ability to reduce or increase the stomatal frequency. So if it has been found that when the CO2 level was very high, the stomatal frequency was low, and when the CO2 level was reduced, then the stomatal frequency was increased. So this plasticity is found in angiosperms, that angiosperm can also change the climatic system, climate, by reducing the number of stomata at the time of high and low CO2 level. So coming to about the plate tectonic history of our globe, that during Triassic time period, we can see that this red area is desertic areas, and the blue is the tropical area, which is confined to the tropical region. So we can see that during Triassic time period, the climate was not very congenial for the growth of angiosperm, because the desertic areas were very high. Even in Torsian time period during Jurassic, this was the desertic areas were too much, and this could be one of the reasons that angiosperm could not diversify. But during early Cretaceous time period, we can see the continents started moving, drifting from each other, and we can see that a lot of angiosperm fossil records we can see in the tropical zone during Aptian time interval. And during Cenomanian time, then when all the continents started moving away from each other, and there is a lot of plate tectonic movement took place during this time period. During Cretaceous time period, we can see that large number of angiosperm records all over the globe. And this also tells us that the climatic condition during this time period was equable type of climate system, like temperature difference between pole and equator was minimum. During Maastrichtian time period, again, we see a large number of angiosperm records. And India was, during Cenomanian time, it was in the southern high latitude. But during Maastrichtian time period, Indian plate started moving towards north, and it was in the subtropical zone at 70 million year time period. At the time of KT, at the time of Cretaceous tertiary boundary interval, Indian plate was just below the equatorial region. And we see a large number of angiosperm records in Deccan Indo-European flora. So there are three major aspects that governs the changes in the continental configuration, and that affects the global climate through three mechanisms. The landmass dispersion due to breakup of one big landmass into smaller continents modifies the source of moisture advection. So like in Gondwana continent was a large big continent during Permian time period, and all the continents were together. So we see that there were period of very humid climate condition, but most of the climate was very arid. Second is the latitudinal positioning of land masses also affected the source of precipitation and hence greatly altered the global climate. And fragmentation of large land masses resulted in the latent heat release and warming of mid to high latitudinal areas. So all these factors, they have affected the climatic condition, and in a way that also helped the angiosperm to evolve. So we can see that during early Cretaceous time period, the angiosperms started evolving, and they diversified by 70 million years. The climate also was very arid during Triassic and Jurassic time period, but became very humid during later part of Cretaceous. So plate tectonic movement also affect the climate in a variety of ways, since ocean and atmospheric circulation are linked to each other, and any process, any change in one changes the other and forces the other, which affects the climate. We know the thermohaline circulation system, that the warm water currents from the tropical region, they go to the polar areas, and from cold water current they come to the tropical region and to cool them. So this is the thermohaline circulation system was also affected because of the plate tectonic movement of the continents. So early angiosperm diversification, we can say that it took place in Gondwana continents, and during this time period, the CO2 was very high. It was an equable climate, lack of polar ice sheet, and during Cretaceous and early Paleogene, that led to the origin of tropical angiosperms. This was the area of origin of angiosperm, as discussed and given by most of the workers. The northern part of Gondwana or Africa, that was the area where the angiosperm appeared, and they diversify, and then later on they were dispersed to other regions. So why was Cretaceous and early Paleogene was so warm, a period of sustained greenhouse Earth? So background condition, we all know the climate was already warm due to massive release of CO2 in Cretaceous time period. And when you see the volcanic activity during different time span, see the volcanic activity of different time span, we see that during compare them with the Paleozoic, Mesozoic, and Cenozoic time period, the maximum volcanic activity took place during Mesozoic time, and that too in Cretaceous. It was a very massive volcanic activity. We all know that Deccan volcanic activity that took place in India. And we have a massive deposit of that activity. So just after the 10 million years gap, 10 million years time period from Cretaceous time period, after the KT, then there is another event known as Paleocene-Eocene thermal maxima, at 55.5 million year at Paleocene boundary. It is also known as super greenhouse period, and it was a extreme warm period, and because of very high CO2 content in the atmosphere. The CO2 level was 6 to 8 times higher than the present day, and this interval took total expansion of this time interval was 200,000 years. So what caused this carbon enrichment? Why was the CO2 level was so high? There are a number of theories on this and this aspect, but one particular theory which is mostly widely accepted is the dissociation of methane hydrate from the ocean bottom. Because the Cretaceous was, it is said that Cretaceous was already warm, and because ocean stagnation was also taking place, and because of the warming of oceans, the ocean, methane hydrate layer at the ocean bottom, they became stabilized, and they release enormous amount of CO2 in the atmosphere. So this was the time period, because this is the time period when the angiosperm, they started coming together and started diversifying and spread all over the globe. So coming to India, where was India? India was, as I just now I told you, that India at the time of KT, India was just below the equatorial zone, but at the time of early Paleogene time interval, at the time of PTM, India was right at the equatorial zone. So, and all of you must be familiar to these places, which is close to Ahmedabad, that these are the desert areas, desert margin and desert areas, the Barmer region, Cambay Basin, and Kutch. So all these areas are extremely rich in lignite mines. So these lignite mines, they are very rich in biota, both plant and animals. So we get a huge number of fossil records from these mines. What not? The vertebrates are there, variety of vertebrates, then large number of pollen spores, and then megafossil, other plants, leaves, stems, roots, insects. So all of them, they provide us some about the past, what kind of biodiversity, what kind of biotope was present at that time in India. If we try to compare them with their living counterparts, that what exactly are these fossil records, they tell us about what kind of biodiversity was present at that time. So we see this type of scenario. These are lemurs, which were once present in India, but now they no longer present in India. They are confined to Madagascar. These are tapis which are found in South America. They were present in India. And apart from that, you see this is a nutmeg family, myristicacea family, that we can say this jitni bhi hamari spices hoti hain, Javitri and Jaifal, which was derived, which was come out from this particular family. It was present in Rajasthan area and Gujarat area. Now all these families, they are confined to in a very small area, that is in Western Ghat area. So there are a number of this biodiversity was so rich at that time, and we get the fossil records that tells us that what kind of vegetation, what kind of plants, what kind of animals they were thriving in India. But because of the climate change later on, they could not survive and they wiped out from India. So this is the study which we have done in 2009. This is the start of this work, tracing the nearest living relatives of early Paleogene fossils. We found that this is in Paleogene time interval, this is from Paleocene to up to Oligocene time interval, we get a lot of pollen records which are present in lignite mines and other in northeastern coal also. And all these fossil records that were present, we tried to compare them with the living counterpart, living representatives, and we could see that these fossils which were present in India at one time, they are found here. This area from 9 degree latitude to 10-degree latitude, where we have the southern Western Ghat area known as the southern Western Ghat area. This area is extremely rich in biodiversity, and it is more or less many planted species are now present as a single species. So monotypic endemic vegetation is also present in this area. So this area shows similarity with our fossil records of 62 to 55 million year time span. So let us see the climatic condition of this area. So if we consider the mean annual rainfall condition, then this area becomes very wet area. This is the Carnatic region. It's more than 5,000 millimeter rainfall falls here. But if we consider the lesser number of dry months, then this area becomes very wet area. This area shows a very wet area. So here the dry months are only 2 to 3. So most of our forms, they show similarity with the living vegetation which is present in this area, and so this is also known as Gondwana relic forest. It has to be conserved because slowly it is going to be degraded soon, and we get a huge loss to our country. So not only Western Ghat, there are many fossil forms that we have found during that time period. They show similarity with the Madagascar living vegetation or African living vegetation. That tells us that all this paleobiodiversity was very different as compared to present day. At that time, those things were present in India. They were not endemic to regions like Madagascar or Africa or Western Ghats. They were present all over the globe in the tropical region, but due to climate change, they are now confined to certain areas. So with that, we started our lignite, our sampling also, and we did a lot of field work in lignite mines in Western India. Then we went to Niveli lignite, we went to Assam, we went to Meghalaya to collect our fossil material. And then we also collected material from the southern Western Ghat and Assam. So where we can get our living representative of our fossil records. So now I'll talk about the paleobiography that when we talk about the evolutionary history of plant and animals, then one particular term, that is paleobiography, becomes very relevant, which is the study of distribution, evolution, and radiation of organisms through space and time. Study of it also provides us clues regarding the evolutionary relationship amongst different organisms. We can also infer age and place of origin of organisms. It helps us in assessing the role of past tectonic and climatic events in the evolution of organisms. There are three fundamental processes in paleobiography: evolution, speciation, extinction, and dispersal. And to carry out paleobiography, the fossil records are required, then their current distributions are required. So current distribution of nearest living relatives of these fossil records, and we also need the dated phylogenetic tree. So for studying the evolutionary relationship, evolutionary study, so one particular aspect is very important, that is the phylogenetic study. The phylogenetic method or phylogenetic tree is a branching of tree showing the evolutionary relationship among various species which are closely or distantly related to each other, and because of the difference in the morphological as well as their genetic characters. So we can do the phylogenetic study based on the genetic material known as molecular phylogeny, and when we do the fossil data, then we can use the morphological data only. So as just to explain you that this is the living species A, B, C, D, E, And based on the similarity and differences in the characters, they are grouped into and known as clade 1. And then C and D, they are closely related to each other. They are in clade 2. So the time of their divergence, when the C and D, they were separating each other, this is the time of we can determine through phylogenetic analysis. We can also determine the divergence age of clade 1 and 2, and also we can also determine the time of or origin of this complete clade. And this takes a lot of time, and a lot of softwares are required to carry out this analysis. And the length of these branches, they provide the ages. So coming back to this, the use of this, using the molecular data and fossil data, now it is being used for predicting speciation, extinction, and impact of climate on diversification of flora and fauna. So earlier, many people were doing only on molecular aspect, and people were doing only on fossil aspects. But when we combine these two, then we get a holistic approach. And this holistic approach of integrating multiple disciplines, ranging from paleobiology to molecular biology, would address the issues of impact of climate change on a species distribution and their habitats, phylogenetic relationship of extinct and extant lineages, predicting speciation and extinction in response to historical processes, conservation measures of a species by taking into account of evolutionary and ecological aspects. So fossils are very useful, and fossils, they provide age to the phylogenetic tree. And with this, by doing this analysis, we can predict the speciation and extinction of fauna and flora in response to past geological events. We can also predict the impact of past climate on diversification of flora and fauna. So I'll talk about some case histories which we have done recently. So let me talk about the evolutionary history of Dipterocarpaceae. Dipterocarpaceae is a family, Dipterocarpaceae or Sal family. It is found in both in deciduous and evergreen forests. It consists of about 580 species and dominates in the South Asian rainforests. So in Southeast Asia rainforest, the Dipterocarpaceae, about 80% of the forest cover is dominated by Dipterocarpaceae. But there are some found in India also, Sri Lanka also, then in Africa also, Madagascar, Seychelles, and South America. So the family showed this jump distribution. So when we see the species, how many species are present in which continent, there is only one species is present in South America, about 2 species in Africa, about 1 species in Madagascar, 1 in Seychelles, 10 in Sri Lanka, 7 in India, and huge number in Southeast Asia. So obvious explanation is that this particular family, and the earlier worker also stated that they also said that this family originated in Southeast Asia, and from Southeast Asia it migrated to India, and and later on then to other places also. But now here the story starts, that we found a earliest record of this family in Deccan Intertrapian deposit also in early Paleogene time interval, and also in Africa. So these are some fossil records, which are pollen fossil records, which are found in Sudan, in Africa. These are the Indian fossil records, which we get in Paleocene time interval, and you see the similarity on these fossil records. Then we have a large number of pollen which we have found in both Rajasthan and Gujarat area, and they are telling us that this family, that most of the forms which are confined to Africa and Madagascar and Seychelles, they were present in India during that time period. So these are all the fossils which we have found, and very detailed study, morphological studies has been done to carry out the phylogenetic tree. So this is the Veteriopsis, genus Veteriopsis, which is found in Seychelles only. It's endemic in Seychelles. And these are from Southeast Asia or the Shoria forests. What you see that it is, but they were present very huge number in India during that time period. So when we do the phylogenetic analysis, this is the method we use. We take out the different characters of pollen. We have, in this case, it is pollen. So all the characters, they were mapped and they were given the character states 0, 1, and 2 based on the primitive and advanced character. More the primitive character, the less number is given. The more advanced character given a higher number. So these character matrix, they were mapped for all the fossil species and living genus of this family, and this character matrix was developed. And then we also taken the molecular data from the gene banks. The people, they have done the molecular phylogeny of this family, and by clubbing that we get this phylogenetic tree. And this tells us that this family originated in seasonal climate of Africa around 102 million years. The family dispersed from Africa to India via Kholistan-Ladakh arc around 70 million years. And most of these forms, they diversify and they had roots in Africa, but they diversify in the very humid climate of Indian plate during its brief isolation period in Eocene. As we all know that Indian plate was few millions of years. Indian plate was confined to the equatorial zone and at the same time, the Paleocene-Eocene thermal maxima even took place. So the family migrated from India to Southeast Asia post-India-Asia collision, where it completely adapted to wet and humid climatic conditions. And the rapid boom in the diversification of the family took place around 20 million years in Southeast Asia, where it gave rise to a very huge Southeast Asian tropical lowland rainforest. This is the history of this family that when Indian plate was started moving towards north, it came in contact with the already present small island chains. These island chains, they were already present and they were also connected with Africa. So when Indian plate first came in contact with these island chains, and through these island chains, many of these tropical forms like Dipterocarpaceae and other forms also, they migrated from Africa to India. And later on, when Indian plate collided with Asian plate, these island chains, they were subjected beneath the collision zone. Not only the plant, there are animal records also. They also tells us that this is the way that many of the tropical forms from Africa, they migrated to Indian plate. So Indian plate was like a ferry that was taking all the tropical forms, and then after the collision with Asia, then transferring it to Asia, and there you see their major diversification now. So this is how that Indian plate was in contact with these island chains, and then later on, these chains, they were subducted, they were consumed by the India-Asia collision zone. And then later on that these forms, they were transferred to Southeast Asia. So this work was published this January. And this was published in Science. Mahi Bansal, my PhD student, she is the main worker. Robert Morley, he is the worker who is studying the evolutionary history of tropical angiosperms for so many years. He was also involved. Shiva Prakash, he is a scientist with conservatory, and so he was also part of this. Surendu Datta, he provided us the amber fossil of Dipterocarpaceae from Paleogene time interval from India. So with this, the enigma of this origin of Dipterocarpaceae has been solved. Another family we talk about, the Abenaceae family. The Abenaceae is a family which is a pan-tropical family. It is mostly that is a timber producing. You must be knowing about the ebony wood, which is excellent for furniture. And this family is it has a 500 species and is distributed all over the tropic and subtropical region of the New and Old World. But we get very little fossil record of this family. But we got the records from again your Rajasthan and Gujarat area. We get all the records of this family, and that these are the fossil records that is extremely rich fossil records. And these are some of the forms which are no longer present in India right now. They are present in Africa only. So again, we have done how the pollen character evolved in this family. This type of work has been done by Mahi Bansal. And then we have done the character mapping and all that. Then we have found this family is originated around 106 million years in Africa. So apart from that, again, there is some work has been done. This is again the evolutionary history of family Ericaceae. There's one particular tribe in Mauritania that provides us several forms of palm kind of fossils. As you can see, these are the fossils. These are the living forms. They are present, and this family is now confined to South America only. So what is the reason? Because in India we get this records of this family, and it was till Eocene time interval it was present in India. After the Eocene, we don't see its record in India, and this could be one of the reasons that why this family could not be transferred to Southeast Asia. And when we see that during Cretaceous time period, it was present in South America and Africa. During Paleocene time interval, again, it was transferred to India. But during late Eocene, till late Eocene, it was completely wiped out from India, and from Africa also. It was started disappear from Africa also, and now it is confined to South America. So this way, the evolutionary history also traces in the deep time that how the family originated, how it diversified, how it was dispersed to different areas, and how it got extinct from different regions. So coming back to my earlier work, that we have found the grass phytoliths. The phytoliths are silica bodies which are found in every plant, but in grasses they have a very distinct shape. So based on their shape and character, we could identify the grasses were present in India at 65 million year time span. So these fossil records we have found in dinosaur dung. And so there is again there is a hypothesis, because during this time period, during late Cretaceous time period, there was one particular mammal, small, which was a small rat-size mammal known as Gondwana mammals. This Gondwana mammals was found in every Gondwana continents, and at the time of during late Cretaceous time period. In India also, we get in from Naskal area and Kisalpuri area. These are the areas of Deccan Intertrapian localities. So we are the most significant thing is that these particular fossils, this animal fossil, they have teeth, which are for eating a very abrasive food material. So the question was, because these they were reported much earlier in the long back in 80s or 90s. So it was considered like why these animals they evolve. And because at that time there were no traces of evolution of grasses, but after the grasses, we found grasses in the same material where we are getting these Gondwana mammals. So with that, we can say the co-evolutionary interaction between grasses and grazes may have greater antiquity than previously believed. Again, this next work which we have, again, this work was done by us by long back, and we have found the earliest record of wild rice. As you all know, there is a lot of debate going on between India and China regarding the domestication of rice. We can't say that domestication of rice took place in India. Probably this study has to be done properly so that we can also prove that. But we can very emphatically, we can say that wild rice was present in India, and from India, it has gone to China after the India-Asia collision. And we can say that while rice evolved in India, or probably it was evolved in Africa, and then from Africa it has come to India, but definitely it was a proliferation and its expansion took place in Indian sediments. So discovery of rice phytolith not only pushes the clock back on the evolution of rice crop, but also that India could be the place of its origin more than 65 million years ago. And at this time, there was no contact between India and Asia. It was the pre-collision time period. So I think with this, I'm going to end my talk. And these are my young colleagues, and so they all are working on plant biology. The students Mahi Bansal, Kajal Chandra, Samiksha, Shalimar, Harshita Bhatta, they are research scholars, and Anumeya Shukla and Gaurav Srivastava, they are scientists. So it's a very small group, and I hope in future we are going to increase the number of people also. So before that, I would like to talk about that what we are doing in our institute. We are working on evolutionary aspects. We are doing the biostratigraphy, paleobiography, just now I told you, paleoclimatic study, paleoecology, paleoenvironment, paleomagnetism, environmental magnetism. We have a very nice paleontology lab, both vertebrate and invertebrate. And we do the geochemistry also, and elemental TLOSL lab. We have a carbon dating lab, which is very old. Elemental, inorganic and unstable isotope geochemistry lab, organic geochemistry, petrology, archaeobotany, dendrochronology, industrial palynology, and ancient DNA, which is a very recent lab. Apart from that, we also do consultancy services in various areas and involving these techniques. So with this, I end my talk, and thank you so much.
Thank you so much, Dr. Vandana ji, for such a nice talk. In fact, this evolution of angiosperm is supposed to be very challenging and controversial topic, but you have narrated and explained to us in a very simple language. And the entire spectrum of evolution you have very nicely told. So we are really very thankful to you for this talk. And now I'll request my colleague, Dr. Vineet Goswami, to please conduct the question and answer session.
Yes,
Dr. Vineet?
Yes, thank you very much, Deshpandeji, and thank you very much, Professor Vandana Prasad, for such a nice talk on the evolution of angiosperms. And so we will start the question and answer session now. So at first, I will ask my colleagues on the WebEx platform to raise their hands and ask their questions to you directly. So I can see some hands raised here. So Professor Banerjee, can you ask your question?
Yes, thank you, Professor Vandana, for your presentation today. I had a specific question regarding one slide where you showed that at 130 million year, the carbon dioxide levels were much higher. So I was interested whether it is possible to know the concentration of carbon dioxide in the atmosphere at 130 million year or even earlier in the past, and how do they compare with the present levels, which are well certified by NOAA, for example, at about 420 ppm. So how much was the average global increase in temperature as well? So these are some of my questions. And thank you again for your presentation. lucid presentation.
So thank you, Dr. Banerjee. And it was because this is this particular time period, Paleocene thermal maxima, even that took place at Paleocene boundary interval. It is now being studied all over the globe, and we see the people, they have done the quantification, and according to these studies, and this is also like negative carbon isotopic excursion peaks. They are found in during at the time of this event. So in India also we get the records of this particular event, and so we have not quantified that how much CO2 level was at that time. This was done by people, those who are in doing work on the northern hemisphere. There are a huge number of localities, they are present in northern hemisphere, and so these people have done it. But we have definitely we have found a negative isotopic excursion peak in some of our sedimentary records in Meghalaya area. And because this particular time interval after the ETM, then there is a ETM2. So ETM2 is the Eocene thermal maxima event, which has been recorded in Bastan area, and we see there is a change in the biological community, and the seasonality we see in the not in the in any other type of change. We see there is a change in the seasonality pattern. The plants which are adapted to seasonal climate, they became aseasonal, like very less number of dry months at that time, this particular event. And so this is what we are doing, and we are trying to see that what type of community before the PTM event and what type of community was present after the event.
Thank you. Thank you.
Dr. Vandana ji, may I request you to kindly switch on your video, because our YouTube transmission is not showing your video, and even we can't see your video. So
Is
maybe
it
your
okay?
camera. Not yet. We are not able to see actually.
Okay, let me, I think I've shared this. It's okay?
No, actually your presentation is there, but your own camera is not there. We are not able to see you actually.
To see me?
Right.
Okay,
So
okay.
your video must be off actually.
Okay. I think video must be off.
You have to.
I have to stop sharing my slide, then can.
You can. Yeah, you may unshare. You may unshare, not a problem. But yeah, now we can see you. Yes, that's right. That's right. Right. Okay.
Yeah. So we will continue the question and answer session. So next question is by Dr. Dvijesh. So Dvijesh, can you ask your question directly?
Yeah, Dr. Vandana, it's a nice talk. It's very informative. So I just wanted to know that you have discussed the role of angiosperm extinction in a particular geological boundary, Cretaceous Paleogene. So can you use this template for other extinction as well, or it is only applicable in this particular boundary?
See that KT boundary, when we talk about KT boundary, everybody talks about KT boundary. It was an extinction event took place, but we don't find any extinction in angiosperms that time period. It was the extinction of mostly in the calcareous forms which are found in ocean, marine organisms. So mostly the extinction that took place because of the very high CO2 content, probably from high CO2 in the atmosphere, the ocean acidification took place, and that resulted in the wiping out the calcareous forms in the ocean. So, but angiosperm, we have not seen any extinction during KT time period, first of all. Second is that what you're saying that this can be used, the extinction took place because of the climatic condition in the land areas. So climatic condition is either the precipitation was not there, or it is very high precipitation that changed the complete community, or it is a very dry climatic condition they started happening. And what we feel is that after the Himalayas were uplifted and the monsoonal type of climate started appearing in India, and because and seasonality started, and those certain areas become very dry. So this has resulted in the wiping out or extinction of many of the forms which were present earlier. So this could be one of the reasons for that. So at the KT time period, we don't see any extinction of angiosperms.
Okay, just a small query that how old is the fossil record, oldest fossil record of angiosperm? Then we can use some oldest stratigraphic boundary, otherwise we can't.
So oldest records are that, as I told you, it's the most authentic records of angiosperms of fossils. They started at about 130, 120 million year time period in Cretaceous time period.
Okay.
And
Okay.
that is the, yeah.
Thank you. Then we can't use in Permian Triassic boundary or some other boundaries because
Yes.
it is dependent. Okay, thank you very much.
Okay, so we will move forward. So next question is by Dr. Manas Samad. So Manas, you can ask your question. You are muted, so unmute yourself.
Yeah, thank you, Vandana ji, for such a nice talk. My question is, one of your slides you talked that one of the angiosperm, Dipterocarpsy, that found in Africa, then moved to India via Ladakh. Okay, and you told us that it might be like 120 million year old when it was in Africa, and kind of 70 million old when it is found in India. My question is how difficult or how easy it is to tag an age to any angiosperm.
You mean to say how we provide the age to these?
Yes.
So age is not provided by these fossils. Age is provided by other biostratigraphy, what we do, that presence of calcareous nanofossils, nanoflagellates, foraminifera, that provides age to the sedimentary successions. And those successions which are already being dated, we then study the fossil records on those sediments. So the age framework is not provided by these fossils because they are not age marker. We are studying them. So age is being given by the biostratigraphy of markers, which are dinoflagellate, foraminifera, and nanofossils.
Okay, thank you. Because why I asked, because we do study stars and we actually tag age to a star, and we found it's very difficult because due to wide variety of stars and we have a lot of difficulties. So I just thought to how you do that. Okay. Thank you.
Okay. Yeah. So Deshpande ji, you can ask your question directly.
Yeah. Thank you. Vandana ji, I have a couple of questions.
Yes.
My first question is that you mentioned about this stomatal frequency. So in response to CO2 concentration, the number of stomatal openings increase or decrease. That is what you mentioned. So in that context, I would like to know that are there any botanical evidences which tell us something about the stomatal frequency variation in response to post-industrial CO2 variation? Are there some recent studies?
Yes, because a lot of people now they are working on different aspects, and those who are working in the biological labs, they have under controlled experiment condition. They have done these experiments, and they have found the plant respond very well with the CO2 content, and the stomatal frequency, they increase or decrease according to changes in the CO2 content in the atmosphere. But it also takes a lot of time. It is not like one day the stomata will appear or next day it will decrease. It takes about years that if this thing continue for long period, then the stomatal frequency also changes.
So is there some hypothesis which tells that if the CO2 increase is there, how it will, how the vegetation will respond?
Yes, there are certain plants where the evapotranspiration rates are very high. The particular plants which takes up a lot of water from the soil, and they evaporate also. There are certain plants which are not like succulents, and they don't evaporate much. So there are a lot of this kind of data is available. And they tell us that the plant community, of course, it will change from one place to another according to changes in the climatic condition or CO2 content in the atmosphere. The CO2 content, if it is not exactly the CO2 content, because of the very humidity factor, and the precipitation is becoming less in certain areas because of CO2 and global warming, that is affecting the plant. Plant can take up CO2 and can modify and can do the photosynthesis, but the problem is that if the heat is very high, the plant respond very drastically, and they are not being able to like consume and then do their nourishment and all that.
So my second question is about the origin of angiosperm. If I understood correctly, you mentioned that the angiosperms actually originated in the northern part of Gondwana, and they spread across. Is that right? So was there
Yes.
something conspicuous about the northern part of the Gondwana? Why they arose there?
Yes, because northern part of Gondwana, that was, if you see the plate tectonic movement of continents, you see that the northern part of Gondwana, that is Africa and south part of South America, they were for a very huge time span, millions of years, they were confined to the tropical region, and that could be the reason that there the evolution, when the climate became humid and slightly humid, the evolution of angiosperms started there. So that is why because Africa is a big continent, within Africa itself, you can see that tropical regions and then temperate areas, and also, so this is the Africa could be the place for many plant and animal species, and from there it migrated to different places. I'm talking about tropical entities. Of course, the temperate flora and fauna, they have evolved in once this ice sheets were formed in both the polar regions. Himalayas were uplifted. Then the temperate flora and fauna that later on they came into picture.
Okay.
So we can also say that the tropical angiosperm, the tropical plants, they are more ancient as compared to temperate flora and fauna.
Okay. And my next question is about the angiosperm response in the PETM. You mentioned that during PETM, because of the warming, CO2 liberated from the ocean. So how angiosperm responded to it during PETM?
For last several years, we are working on this problem and trying to find out that in the low land areas, or in the low latitudinal areas like tropical areas, the effect of PETM in tropical region. We are trying to find out, because we cannot compare our data with the data which is coming out from high latitudinal areas in northern hemisphere. Most of the work has been done in northern hemisphere, and there it is so easy to, because they see a very drastic change in the community. When the PTM arose, that when PTM occurred, that at the time there is a very different, the flora completely wiped out, completely changed from the type which was present earlier. But in India, we don't see this kind of very drastic change. We just see that there is a diversity pattern which is started because we are in the tropical region, and in tropical area, because the plants, they are mostly tropical plants, and they are mostly governed by the precipitation. So the precipitation data is now tells us that it was a seasonal climate. So the plants, they were before the PTM, mostly the plants, they were present, they were having a very adapted to a seasonal climatic condition. There was some kind of season precipitation of this period, so 6 months of precipitation period. And then after the PTM, or at the time of PTM and post PTM, then we see that there is a change in the flora, and the flora is more adapted to a very humid climatic condition. And it was it shows that during that time period, the dry months were very minimum, about 2 to 3 months of dry period. So this is what we have seen, that there is but we can't have that kind of very dramatic kind of change what in northern hemisphere, in high latitudinal areas, people are seeing. It's a very subtle change, and you can see that there is a seasonality is changed during that time period.
I have one more question. That this, you mentioned very curious observation that these spices were growing in Gujarat and Rajasthan, which have now migrated in the Western Ghats. Did I understand correctly? Did you say so?
Yes, these spices, they come from a very humid forest, and which is tropical rainforest, which is confined to southern Western Ghat area. And so it is called Myristica swamp. You all must be knowing about Myristica swamp, which is a habitat, and which is we take care of a lot of plant and animals in this swamp area. So Myristicaceae family is a nutmeg family. It provides us this all the spices, jaifal, javitri type, and so this particular family is now confined to Western Ghat area. But at that time, it was present in Rajasthan and Gujarat area. So one can imagine Rajasthan and Gujarat area, how wet they were.
Okay. And my last question, it is not a scientific question, but you mentioned about the consultancy services provided by BSIP, and then you listed the instrumental setups also. So will you please comment briefly that what exactly is the consultancy services you explained?
So we are recently, because government is giving us very little fund for the, because our institute is basically designed to carry out this basic research, but now we are doing a lot of work for oil exploration in oil exploration areas, and we are working with ONGC. We have 2 projects from ONGC, and we are doing biostratigraphy, paleobiographic mapping of Cambay Basin. Then we are also doing pre-Cambrian this area. That is, we are also doing oil exploration work is also going on there. We are providing the biostratigraphic data, paleo environment, and the source rock character. So this is what we are doing. We are also doing some consultancy and for doing the geochemistry. There are many people, they come for organic geochemistry or inorganic geochemistry. And C14 dating is a very old lab from our institute. And we also do a lot of consultancy services from this lab. We have confocal laser microscope. We have SEM. So it's a very small, of course, it's not such a big thing, but definitely we are entering in 2 areas. One is the oil exploration area. Second is the characterization of coal. So this is in 2 areas we are entering now.
Thank you very much, Vandana ji, for answering my questions. Thank you, Vinit.
Okay. So we have a question from YouTube. So it is asked by Dr. Shashi Kiran Ganesh. So he would like to ask, now that amino acids have been seen on asteroid samples brought back to Earth, what do you think of the theory that life arrived on the early Earth by asteroids or comets? That is the panspermia hypothesis.
I can't comment on this aspect. This is you people have to take care of. Yeah, of course, because there are a number of theories are now being given that how life evolved, I think the answer can be with the those who are working on Archean and Precambrian time period, because earlier they were only studying the just morphological aspects, but now people are doing the chemistry side of during that time period. So they can provide some clues regarding. I have absolutely no idea about that amino acids present in the Mars and those areas. Definitely, it's a very upcoming area. And we can, BSIP can also provide, because we were actually thinking with a lot of data, people are, those who are working in Precambrian time period or Archean working, they are providing us very significant clues regarding the early life, the chemistry of early life. So that can be utilized and I think can be combined with the people who are working on astrobiology.
Okay. So I don't see any other questions from the YouTube, and and I don't see any hands raised on WebEx as well. So I would like to make a last call if somebody would like to ask the question to our today's speaker. Okay. So if not, I would like to ask one question before we just close the. So it is related to, because you mentioned that angiosperms, they were not affected through the KPG or KT transition, but there were extensive wild fires subsequent to the KPG event, KPG boundary. So what, they were not extinct, but their abundance, was it affected in some way after subsequent to the Cretaceous Paleogene event?
See that KT boundary interval, or we can say the volcanic activity that took place in India that time period, but this volcanic activity was not continuous. It was in phases. So during those phases when the volcanic activity was taking place, at that time the plants were affected. But after after say about thousands of years or so many time lapse after that, then the life started appearing, and then because all the areas were not affected by Deccan volcanism, so those areas which were not affected by Deccan volcanism, there the life was continuing, there the plants were continuing, so they were like dispersed from one area to another area. Second thing is that the volcanic rocks are extremely rich in nutrient. So this nutrient has also given us given the angiosperm to like diversify or giving a lot of nutrient to plants which were developing and were present at that time on Indian plate. So at KT time interval, we don't see any land extinction, land plant extinction. The extinction, what we see is the organism which were thriving in the ocean because of the change in the ocean chemistry. Those animals, those planktons, they were affected. So this is what we are actually found out that this is the KT boundary, because that's a lot of people are talking to us and they say that why don't you know talking about the extinction, but we have not found any extinction in plants.
Okay, just one connected question. So was there any evolutionary change in them subsequent?
Yes, evolutionary change in them is there, because I don't know whether this was because of the movement of Indian plate from southern mid-latitude to equatorial region, because it is so difficult to decouple all these effects. Indian plate is moving, climate is also changing, PTM is also happening, KT is also happening. So it is really difficult to decouple all these effects. But definitely the latitudinal position of India, because Indian plate was moving very fast at that time, and it was in the subtropical zone below the equatorial zone at the time of KT when it started, and at the time of post-KT, Indian plate was coming in contact with the tropical zone. So there definitely the climatic conditions where they change according to this. And then later on, because of the PTM again, then there was an Indian plate was right at the equatorial zone. The climatic condition became very humid and very, very wet.
Okay. So thank you very much, Professor Vandana Prasad, for answering all of our audience's questions so nicely. So that ends or brings conclusion to our question and answer session. And I would like to invite now Professor Kuljit Marhas for giving the vote of thanks.
Thank you, Dr. Vineet. I, Kuljit, on behalf of Amrut Vyakhyaan team, would like to thank our today's speaker, Dr. Vandana Prasad, for such a fascinating and informative 45th Amrit Vyakhyaan she delivered, and for initiating such a lively discussion within our members. I would also like to thank our director, PRL Director, Dr. Anil Bharadwaj, and PRL Dean, Dr. Pallam Raju, for supporting AV team always. Last but not the least, thanks to all the members who are joining us on WebEx as well as on YouTube and initiating the discussions. I hope with such enthusiasm you will join again next week, same time for next AV. Thank you so much.
Thank you. Thank you, everybody. Thank you.
Thank you. Thank you very much.
Thank you.